All. This is Dr. Mobeen Sayed from drbeen.com. Welcome to one more show. So the study that we are going to discuss today is actually by researchers from Brigham and Women University or Hospital from Boston, Massachusetts, Harvard Medical School, Boston, Massachusetts, and the Division of Infectious Diseases, Montreal University, Quebec, Canada. Beautiful study. The summary of the study is the following. Individuals, these were healthcare workers in the study, 13 people, so small study. Individuals who got the first shot of Moderna vaccine, they had spike protein, some of them, 3 of them, but all of them had S1 part of the spike protein in their plasma. That was not supposed to happen, and that is what is observed, number one. Number two, this is not a conspiracy theory. This is also the first ever study to show vaccine-generated S1 or spike proteins part circulating in the blood. So I would show you, as part of the discussion today, these texts in the study that show that this is the very first ever demonstration of S1. The researchers feel, so they use a specific test that they are inventors of as well, I believe. So the researchers say that possibly other researchers have not been able to see S1 or spike in the blood because the other test kits are not as sensitive. However, because they have a conflict of interest, the Brigham and Women Hospital took part in resolving that conflict and making sure that the study is not biased because of that conflict. So this is the summary. Let's start our discussion. There is an interesting part of the study, and that is that I believe that researchers stopped short of explaining the possible outcomes of finding these proteins in the plasma or presence of these proteins in the plasma. So I have also some extra studies here that would demonstrate what can be a potential problem with these spikes and S1 in the plasma. And my thinking that why they stopped short, number one, maybe this was what their primary goal was, to see if there is spike and S1 present in the plasma or not. And maybe also that their funding and their resources were not sufficient. So we'll see. So starting our discussion, this is drbeen.com. If you would like to have access to my premium courses, then you can go to drbeen.com. In the description of this video, there is a link that offers the premium access at a price that you would not have seen. I was trying to calculate it yesterday that if you have access for 3 years, it is actually lifetime access. But if you use it for 3 years, it's actually not even $1 per month. Okay, so with that, here is the study. Clinical Infectious Diseases, Oxford Academic, that is where it is printed. It is published Feb 15, 2022. Although it was accepted and published in last year, but here, this is the journal issue in which it is printed. This is another study talking about ACE2 spike protein, and when spike protein binds with ACE2, the imbalance that creates for the inflammation and anti-inflammation, pro-inflammation and anti-inflammation system in our body. This is another study that talks about S1 protein, if present, can cross blood-brain barrier and what kind of an outcome that can cause. And remember, so far, the thought was that there is no S1 protein in the plasma. There is no spike in the plasma, especially from the vaccines. So these were all experimental studies where S1 was injected in the plasma of the animal and then observed the results observed. This is another study about the S1. We'll talk about this. This study says that S1 or spike in the plasma can cause hypercoagulability, and that may be important to understand why some people become hypercoagulable with SARS-CoV-2. And this one, I believe we have done this discussion before as well. This is Dr. Bruce Patterson and his study about S1 present in the monocytes of the COVID patients. So with these studies in our context, let's start our information, our discussion. Here is the PDF, and I have highlighted some parts. We'll look into that. Let's look at my drawings. So we'll start from here. These are our gifts for humanity continuing. This is the summary of the whole thing. The summary is, and I've taken this excerpt from the study, these data show, so this is authors writing, these data show that S1 antigen production after the initial vaccination can be detected by day 1 and is present beyond the site of injection, that is deltoid, and the associated regional lymph nodes. So what will happen is, if you can see my deltoid here, so when the injection is given to the deltoid, the antigen from here, or the broken cells, the whole structure here that is taking part in the vaccine activity, those antigens will be carried through the lymph to the local lymph nodes, and that could be axillary lymph nodes, that could be the cervical lymph nodes here. So there may be actually swelling. Some people would say, hey, my lymph nodes got swollen in the armpit or in the neck. So the researchers are saying that finding the spike proteins or S1s in these areas may be okay, because that is where these antigens are going to go, and that is where the immune responses are occurring. However, they have found that on the day 1, the S1 is present beyond the site of injection and the associated local lymph nodes. That is the most important thing. And that is what they're saying. This is the first ever. And let me show it to you. So here, this is the study. Although in the journal I showed you, it was printed there or published there on Feb 15. Originally, it was received on 6 April 2021. It was the editorial decision to publish that, accept that, and publish that was 13 May 2021. It was published online on 20 May 2021. Okay, so read this with me. Nonetheless, this is also from an excerpt from the study. Evidence of systemic detection of spike and S1 protein production from the messenger RNA 1273, so this was Moderna, vaccine is significant and has not yet been described in any vaccine study. This is the importance of this study. This is not any other willy-nilly study that is just creating a conspiracy theory based on some conjectures. This is a decent, good study. And they're saying that this kind of a study has not been done before. This presence of spike and S1 has not been demonstrated in the plasma before by any vaccine study. And they say, why? Why? Likely due to limitations in assay sensitivity and timing assessments. So now let's look at the summary of the study. Small study, 13 healthcare workers, young age, 18 to 30 years. And they say that this may be one of the limitations of this study, that the individuals taking part in it are more on the young side. So they took their plasma when they got vaccinated, day 1 and so on. So they had multiple samples for the first 28 days. Then after they got the second dose, then they had once again multiple samples after that too. And what they found was that after the first dose, which was 100 microgram of mRNA 1273 by Moderna, they saw that S1 proteins were produced on day 1. They were present in the plasma. S1 present in the plasma, day 1. Then they saw about day 5. In some, not all, S1 was present in all. On day 5, in 3 participants, the whole spike protein was in the plasma as well. And they discuss in their paper, they say one reason that we may not have seen it in all may be because their test kit is not able to detect a cleaved or broken spike protein. So if the spike protein entered the blood, but before entering or after entering, it got broken up, then their test kit cannot figure it out. The second shortcoming of their test kit is that if the spike protein is now bound with the antibodies, then that complex is not detectable by these test kits. So there may be spike and antibody together running around in the blood, but that may not be detectable. And of course, after the first injection, if the person was not previously exposed, then having antibodies is not going to be possible because it would take some time for the body to make antibodies. So what is the first message here? First injection of Moderna, the very first day, the day 1, spike protein are in the blood. The S1 are in the blood. Day 5th, spike protein can be seen as well for some. So then the question is, why? This time today, I'm not going over every single part of the study. Instead, I wanted to share this interesting demonstration and why it is happening and possibly what does it mean. So question, why is spike protein in the blood? So they have a conjecture. This is the same that I had been discussing in the past. And that is, imagine this is a cell, this blue one. This is a muscle cell, although they don't look like this. Muscle cell or a fibroblast or some other cells in the local area of injection. The lipid nanoparticle entered into this cell. Inside the cell, the spike protein is made, then it is broken down as well, and the antigens are presented on the surface of the cell. We've done this discussion many times. Once the antigens are present on the surface of the cell, then our immune system will become active against this cell. And what would happen is, let's say this is a cytotoxic T cell, CD8 positive T cell, or CTL, the killer T cell. And by the way, we saw yesterday in another study that I presented yesterday that natural killer cells, which are part of the innate immune system, can also do this action that cytotoxic T cell would do. So that study is also interesting because that was the first time somebody showed that NK cells are taking part in this whole process of responding to the spikes. So here, this is a CD8 positive cell. It would bind with the antigen presenting cell. And when it would bind there, what it will do is, remember, it has perforins and granzymes. Perforins are like little protein that would create perforations or holes in the target cell. These are the holes here. And the granzymes are like small grenades. These are also proteins that are released in the local tissue fluids, and they would swim and go in to the cell that has perforations or holes in it. The end result is that the cell would die. It would be broken down. So this is one outcome of this cell. So the researchers think that when T cells are killing these cells, then the spikes are spilling out of them. And you could say that this is also possible with the macrophages when they are doing it. or natural killer cells or even neutrophils. Although neutrophils do not directly kill the cells, but neutrophils can pick up the local debris and do similar things with that. Anyways, from the study's point of view, they only talked about CD8 positive or T cells. So this much, they think that they have a hypothesis. The question for them as well, why S1 in the blood? That, they say, is not clear to us. And I have some theories, I would discuss them later. And this is where I feel that it is possible that they could have extended their study to discuss those. But again, it depends upon what was their primary mission with the study, what were their resources. Otherwise, they could have continued to go on for a long time. So anyways, here is blood. And in the blood are S1 parts of the spike protein. They say, we have no idea. It is not clear to us why spike S1 will be in the blood. So I'll discuss some a little later. If you see, this is an excerpt from the study. They say, we hypothesize that the cellular immune responses triggered by the T cell activation, which would occur days after the vaccination, lead to direct killing of the cell presenting spike protein and an additional release of spike into the blood. And they say the mechanism underlying release of S1 and the subsequent detection of the intact spike protein remains unclear and requires further studies. So we don't know. So we do not know why S1. And we also, they say, do not know why more intact spike proteins later on in the blood. So then, before we discuss my links to the other studies, let's go back to what they are saying. They're saying, are these spikes then cleared or not? And so let me show you their graphs, and then I'll show you my graph. So here, if we go to my homework, so here, this A graph over here is the spike S1, B is the spike, and C is the nucleocapsid. They were actually detecting nucleocapsid as well as a control, because after the vaccine, we should not even see these, but anyway, spike or S1, but not nucleocapsid, because nucleocapsid is part of the virus, not of the spike generated by the vaccine. So for sure, they saw that there was no nucleocapsid. And if you see here, day 0, 1, 3, 5, 7, 9, they saw that S1 increased for some days. Spike increased for some days as well. And then as antibodies started appearing, IgG and IgA, then they started clearing the spikes out. I have a point over here from their own message. They said, and this is true for majority of the test kits, they said our test kit cannot identify complexes. Complexes mean that here is the antigen. So let's say this is a spike protein here. S1, S2 spike protein. And then there is an antibody that is bound here. So this is antibody, this is antigen. This whole thing is called a antigen-antibody complex. Many test kits cannot detect this antigen anymore because it is already bound with the antibody. And so these go unnoticed. When they say that, hey, we've gotten clarity after the antibodies are formed, it is also possible that actually there is no clarity, but there are antibodies which are quickly binding to this spike protein. So then your question could become that, well, we're good then, right? We have gotten the antigen and antibody connected with each other. It is essentially equal to spike getting cleared. Maybe. Maybe. But I would actually request you to read up on type 3 hypersensitivity reactions, where this complex, antigen and antibody complex, can get deposited on some tissue. So let me back up for a second. In a normal physiological mechanism, this complex is picked up by macrophages and cleared out. But these complexes are known to get deposited on the surface of the blood vessels, on the surfaces of the joints, on the surfaces of kidney, on the other barrier areas, and cause local inflammations and damage. This can cause vasculitis. Now, tell me this. Have you heard of vasculitis with the vaccines? Yes, we have heard of the inflammation. We have heard of the clottings. These are signs of vasculitis. Have you heard of neurological symptoms? It is entirely possible that these complexes are settled on the blood-brain barrier. They cannot cross the blood-brain barrier, but they can settle there because there is filtration happening. There is pressure there. They can cause inflammation over there. So as much as they can be cleared out by the macrophages, which is the right thing that would happen, these can get deposited on various tissues as well and cause type 3 hypersensitivity reaction. And that is a known reaction. It's not anything unique. So back here, they're saying that yes, when the S1 is present, the spike is present, and after that, as the IgG and IgA, immunoglobulin G and A, are produced, they would clear them out. They also say that we do not see any spike or S1 in the blood after the second injection. But once again, the same thing would occur. After the second injection, it is possible that even when the spikes are coming out in the blood, the antibodies are being produced fast enough as well, because this is the second injection. Our body knows about this situation and can very quickly produce the antibodies. So those complexes will be formed very fast. The test kit does not pick up those complexes. So the point is, it's not necessary that after the second injection, this S1 and spike protein does not spill in the blood. Instead, it is possible that they are very readily formed into complexes. And the authors say it by themselves as well, that their test kit does not pick up complexes. Then the question becomes, what is clinical importance of this all? And this is where I believe, once again, authors stayed short of offering links to the other studies. And they simply said, we do not know what is the clinical outcome. So let me show it to you. I thought this was a little disappointing for me to read, that they simply stood back and did not further discuss it. So let me read it. It is somewhere over here. Here. The clinical relevance of this finding is unknown and should be further explored. So they said, we don't know. So I'm going to connect some dots here, and I wanted to make sure that we have author's position in front of us, so that when I am providing these connections, these are not what authors are saying, this is what I am connecting. So authors say they do not know. Now let's review some other studies. Let's now agree, in light of this study, there is spike or more possibly S1 present in the blood. What can this S1 do? And what can this spike do? The first one, spike, when it binds, so let's say here is a cell and there is this ACE2. We all know this. I don't think this is something unique I'm discussing for the first time. When the spike protein will bind to the ACE2, it will occupy this ACE2. And that would cause an imbalance between pro-inflammatory behavior and anti-inflammatory behavior. pro-inflammatory behavior of ACE1 and anti-inflammatory behavior of ACE2. Now, ACE1 and ACE2 are usually in a balance, but if you occupy the ACE2 with a different molecule, then this ACE2 is not available to work with the normal physiological molecule in our body, and what happens is we go in a pro-inflammatory state. This is a known mechanism from beginning of the pandemic. This is how the inflammatory imbalance occurs. This is one part that tips us towards the cytokine storm. So it doesn't cause the cytokine storm, but it is one part of it all. So if there is spike in the blood, there can be inflammatory outcomes. And we have seen that if not all, then some people who are vaccinated have those injuries or side effects. And let me present that study here as well. Here is the study. ACE2, much more than just a receptor for SARS-CoV-2. So you can go over the study and read this. Second part. S1 can cross blood-brain barrier in mice and cause neurological issues. Why they talk about in mice? The reason is you cannot just take a human being and inject S1 in them and say, okay, I'm going to see if it crosses your blood-brain barrier or not. That's not going to happen. So they do this in mice. So they take S1. They don't take spike protein. They take the S1 part and inject it in the mice. And why we were able to dismiss those studies is to say, well, S1 is not going to be in the blood in normal cases. So yeah, sure, you're showing that a mouse, when injected with S1, that S1 can cross a blood-brain barrier and go to the brain and cause inflammation. But because this normally doesn't happen, your study is interesting but not practical. But here with this study, if the S1 is present in the blood, as the study is showing, and remember that Brigham and Women Hospital was involved in this because there is an author who may have had a conflict of interest. So the hospital was looking at it to make sure that the study is done correctly. And the healthcare workers were from this hospital. So once again, it's not a conspiracy theory study. So here now, S1 can cross the blood-brain barrier. If it can cross that, if we've seen it, maybe it can cross it in the humans as well. So the only question was, well, we don't have S1 in humans. But here is a study that shows S1 after the vaccine can be in the blood. So now let me show you that study. The S1 protein of SARS-CoV-2 crosses the blood-brain barrier in mice. So we show that intravenously injected radioiodinated S1, so that they can trace it, readily crossed the blood-brain barrier in male mice, was taken up by brain regions and entered the parenchymal brain space. S1 was also taken up by lungs, spleen, kidney, and liver. And then down here they say, APOE genotype and sex did not affect the whole brain S1 uptake, but had variable effects on uptake by olfactory bulb. By the olfactory bulb, the nerve for the sense of smell, liver, spleen, and kidney. And the uptake was in hippocampus, which is a part of the brain connected with the olfactory region, plus it's part of the limbic system, and so connected with the emotion and those things. Hippocampus and olfactory bulb was reduced by lipopolysaccharide-induced inflammation, and result was there was inflammation in the brain, and that caused reduction in the olfactory bulb, that is the sense of smell nerve, and the hippocampus that is attached, which is part of the emotional system, and it does many other things too. Mechanistic studies indicated that S1 crosses the blood-brain barrier by adsorptive transcytosis, and that murine angiotensin converting enzyme 2 is involved in brain and lung uptake, but not in kidney, liver, or others. So what they're saying is, the question becomes, how does S1 enter the brain? How would it cross the blood-brain barrier? And the answer was, adsorption. What is that? So blood-brain barrier is a very specified and very mature area. So here in the blood-brain barrier, imagine this is a blood vessel. Inside the blood vessel are the endothelial cells. These are the cells that are making the surface of the blood vessel. Correct? We know that from ever. Then we know that around those are more cells. So it's a properly sealed area. And then around those, so there are pericytes, and then around that is the podocytes or the astrocytes, pods. So if something is going to cross all of this, all of these layers, that crossing is done with adsorption. In the adsorption or transcytosis, what happens is it's not that you pass between the cells. It's not that you go like this. Instead, you actually enter the cell, get absorbed into the cell. A tiny hole appears, and then you are allowed to pass through, to float through, to row through that little hole all the way to the back of the cell, and then from there enter the brain tissue. This is transcytosis and adsorption. This is a specialized mechanism within the blood-brain barrier. Blood-brain barrier has many ways of allowing the the products to get into the brain or out of the brain. There are specialized channels, there is transcytosis, there is adsorption, there are permeabilities as well, and so on. So they say that this is the mechanism. Now, if this mechanism is possible, it is possible that this could happen in humans as well. And this now tells me why some people who develop the vaccine-related neurological outcomes, this could actually be that the S1 is... So we had been saying so far that these are inflammatory molecules that may be crossing into the brain and causing local inflammation inside the brain and activation of the immune system of the brain or glial cells. But it actually may be that the S1 is entering there. Then here, so this is the next one. This is also very important. S1 interacts with platelets and fibrinogen. This is now within the blood, not in the brain, causing hypercoagulation, clotting. Here is that study. This study, once again, they used S1 separately. And what they did was they put that S1 with the platelets and with the plasma that was poor in platelets to see what does S1 do to platelets and what does it do if the platelets are not present. And what did they find? They found that S1 was able to trigger clotting by activating platelets or by activating fibrin. If there are no platelets, it was able to activate the fibrinogen, which would then become part of the clotting. Remember, platelets, the clotting occurs when the platelets start adhering with each other, and then we tie them together with the fibrinogen or fibrin. The fibrin acts like a rope. And then when the clot is broken down, the fibrin is broken down, and that is what D-dimer is. They saw that S1 does that. But the only thing, once again, was that there was no possibility of S1 from vaccine in the blood. So we said, well, the vaccine won't do it. The patient who had SARS-CoV-2, they could have many reasons for the clotting. So here now, we know that clotting can occur with S1, that is one. And now we also know that S1 can be in the blood. So again, the original authors of the study stayed short of saying these things. They simply said, clinical outcome, we don't know. And remember, these were healthy individuals as well. So these individuals, if they had any side effects, they would probably have recorded them. So possibly they got the vaccine and they were fine. I got the vaccine and I was fine, for example. So it's not that everybody who got the vaccine just fell flat and died. But there are some people who had bad outcomes. So here is a very important clue for why this could be happening. And then finally, this big study, Dr. Bruce Patterson, who said that monocytes, which are at the boundaries of the blood vessels, are seen to be containing S1 in SARS-CoV-2. And there is a lot of discussion over here that the monocyte would pick up S1. How would they pick up S1? That is a big problem. So their conjecture was, so let me go there. Where are my highlights? Okay, so my highlights are not here for some reason. Sometimes the software that I use for highlighting, it just forgets the highlights. So one possible explanation for S1 protein in non-classical monocytes could be that pre-existing CD14, low CD16 positive cells could phagocytose virally infected apoptotic endothelial cells with subsequent degradation of the RNA and presentation of the S1 protein. So what they're saying is, it is possible that a cell that has antigen, I'm just going to use this diagram for a second, a cell that had the antigen on it, and that cell got eaten up by the phagocytes, then those phagocytes ended up dead, and then they got eaten up by the monocytes, and now monocyte has the junk and trash that was in the phagocyte in the monocyte as well. And then they, in this study, and I think we're all aware of it, they say that this results in monocytes releasing inflammatory markers, which then causes the long COVID-like symptoms. And so they have specific management approaches and all that. But here we're talking about S1 as well. Again, I'll keep saying it. This is not what authors said. Authors simply said, we do not know about the clinical outcome. So let me show it to you. The clinical relevance of this finding is unknown and should be further explored. So staying sincere to their presentation, I cannot put my words in their mouth. They simply said, do not know. I am connecting these other pieces for us. So going back here, that is the, in my opinion, clinical importance. There is more than simply saying we may not know. Now, question, why is S1 in the blood? Again, authors said it's unknown to us. So this discussion that I'm going to do is not author's discussion. This is me. And so I could be totally wrong. One possibility, so S1 is for sure part of the whole spike protein. So at some point, the spike protein got produced, then it got broken up, and then S1 part is out. Now, it can be broken up within the cell that is producing it, or it could be broken up outside, or it could have bound to the ACE2, and the spike protein got cleaved by TMPRSS2. However, before you say, aha, Mubin is wrong here, I'm going to shut down this video, I'm done. So before that, let me clear it. Normally, when S1 binds with ACE2, it does not unbind. The ACE2 and S1 are recycled together and destroyed together. So having ACE2 in the picture may not be the right answer, because ACE2 would not leave S1 out. At least so far, that's what we know. Maybe tomorrow there is going to be another study that says, well, we had been thinking that ACE2 and S1 are not separable, but here we are proving that they are. So far there is no such study. So then what are the possibilities? One possibility is that the cell that is vaccinated, that cell is producing spikes, and within the cell, endosome is shredding the spikes. And so maybe that is where the S1 is produced. And when that cell is killed, not only spike is spilled, but S1 is spilled as well. That's one possibility. The other possibility is that, let's say the whole spike is spilled, but then that spike is picked up by phagocytes, neutrophils. Neutrophils are like bad behaving cells. They would eat up an antigen, shred it, and then just release it in the local tissue. Why do they do that? They are actually intentionally releasing that trash so that the lymph would carry it to the lymph node, and the lymph node would respond. The cells in the lymph node would respond. So they're actually doing a good thing. Anyways, so it could be that neutrophils are releasing pieces of spike protein. It could be after the cleavage, but as I discussed, that is less possible. Then it could also be, as the Bruce Patterson's paper says, that maybe the phagocytes ate up this cell. The cell had the spike protein and S1, then the phagocyte ended up having those, and S1 is sticking out there. Now, how would that phagocyte release it in the blood? I don't know. So the most possible case would be killing of the vaccinated cell, which would then release S1. Now, some questions that are in my mind. Number one, and this is the last part, and then it's 42 minutes, I know. Could some people not make sufficient antibodies to remove the spikes in S1? We know that the response to SARS-CoV-2 is that not everyone makes sufficient amounts of antibodies. And some people actually make less antibodies and do more cytotoxic responses. Is it possible that this spike protein problem may be a problem in some who are not making enough antibodies to clear them out? So they got the vaccine. Ideally, the thought was that antibodies would clear these things out, but antibodies are not produced in the correct amount, or their body responded more with the cytotoxic T cells, which is normal physiological function as well, and now they have spike proteins running around in the body. Could the antigen-antibody complexes give rise to type 3 hypersensitivity reactions in some individuals? So is it possible that some of the lingering side effects are because spikes are being produced, they are being bound to the antibodies, and those complexes are now running around and they're settling on the endothelial surface, causing vasculitis. They're settling on other places, for example, joints, causing local inflammation, causing joint pains, and so on. And then, could S1 have crossed BBB, blood-brain barrier, and this S1 got picked up by monocytes? So these are questions. I don't have the answers to them, because for those answers, we have to actually follow this S1 to figure out where does it actually go and what kind of inflammation it does. Maybe this whole thing is totally innocent and the antibodies come in, they bind with S1 and they clear them out and done. And that may be the reason that in some people, vaccines are not causing side effects, and then in some they do. This is the discussion. And this is a discussion. Thank you very much for listening. I saw a comment and I wanted to read it, but then the comment scrolled away. Thank you very much for watching, for being here. Please do me a favor. If you would like to have access to more of these lectures, if you like these kind of lectures, then get an access to Dr. Bean's premium account. There is a link in the description, and the price is so low that it's not even 2 lunches, and it's only one-time price. It's not even recurring. So take advantage of that. And in that process, you would actually support my work as well. And if you wanted to support this work in other ways, you can use PayPal. That link is in the description as well. Or you can buy me a coffee. Link is in the description as well. Or you can become a patron. Even if it is a patron of $5 per month, it is a valuable support, especially in the days when people are trying to get my financial sides broken. So with this, have a nice weekend. I will see you on the Chit Chat channel in a few minutes, and we'll do some discussion. Maybe we'll make some drawings with the facial expressions, and we'll go from there. And if you're not going to be on the Chit Chat channel, have a nice weekend. And please like, subscribe, and share this video. And see you on Monday. Bye-bye.